So we're going to be doing, I have the original ABO video already posted, but this is additions to that video. So this is like an ABO 2. Oh, give me a second.
Yeah, Dad. 25. Hey, Dad, I'm doing some tutoring right now.
Okay, so this is going to be an ABO 2. ABO. Video. Okay, so first, do y'all know who discovered ABO the ABO blood group system?
Oh, yeah.
Yep, Landsteiner. So Carl Landsteiner discovered the ABO blood group system in the 1900. Now, Landsteiner's law, do y'all know what that is? Landsteiner's law.
Doesn't it have to do something with all the blood groups?
Well, it generally applies to all of them, but healthy individuals possess ABO antibodies to the ABO blood group antigens. Now look at this, and next, is it going to be present or absent from their red blood cells? So look at that and see if I should leave in present or absent. Landsteiner's law, healthy individuals possess ABO antibodies to ABO blood group antigens, either present or absent from their red blood cells.
Present.
So if they have the antigen on their red blood cell, they will make antibodies to it.
No. Absent.
Absent. Yep. So healthy individuals possess the ABO antibodies to the ABO blood group antigens absent from their red blood cells. So if they don't have the antigen, they can make antibodies to it and will possess those antibodies. And now I wanted to add in some information that I had written down and not included in the last video. And it's going to be your ABO blood group system in regards to infants or neonates. So for like an age range here, 5 to 6 weeks gestation. Now, what does gestation mean? At what point is that? Is that 5 to 6 weeks after birth or after conception?
Isn't that after conception?
Exactly. Yeah. So don't get this confused with right after birth. This is right after the child is conceived. So 5 to 6 weeks gestation, which is around the time that you can hear a heartbeat, is when these ABO antigens can be detected in the embryo. And now next is newborn. So how are your antigens going to be developed? Your ABO antigens, how are they developed in a newborn? Are they strong? Are they weak? Are they?
Wouldn't they be weak because they're still developing?
Yes, they are weak or partial. So they're weak or partially developed antigens. Now next I'm going to put 2 to 4 years. And that will be about the time of full expression. And then last is going to be 5 to 10 years. 5 to 10 years. And at this point, that's where you will have your max titers for antigens. And after that, they'll decrease with age. So max titers. And then will decrease with age.
So what will decrease the older you get?
Your antigen expression.
So the older we get, the more we lose our antigen?
Correct. The weaker they get, the weaker your reactions get.
Oh, that makes sense. Because if you're elderly, then that would be reason why your immune and all that would go down, is because you don't have. I don't understand that.
That's a little different than the antigens. They just, as you get older, they get weaker, just like starting off. So your immune system goes down too when you get older, but it's a separate mechanism than with your ABO antigens.
So this is just saying the older you get when you're younger, your antigens decrease because you're building up an immune system that helps fight.
Not quite. So your immune system does have to do with like the kind of reaction you would get, like with a transfusion reaction or something like that. But what this is saying isn't so much about your ability to make antibodies or your reaction to other antigens, alloantigens or auto antibodies and stuff. But what it's saying is that the expression itself, like the actual antigens on the surface of the red blood cells, weaken, decrease or weaken.
Okay.
So it's separate than your body's immune response.
Okay. All right.
However, it does weaken too. But it's saying it's weak. Like if you're forward typing them, that really doesn't have to do with your body having a reaction or your immune system, abilities at that point, but it's still decreased. So there's 2 major groups with decreased expression. And what are those?
The elderly and the infants, I guess.
And the infants. Yep, so the old and the young. So now what does that mean for you in the lab? If you have all negative results for anti-A, anti-B, and then A1 cells and B cells with a negative autocontrol, that might be a group O newborn or elderly patient. So I'm going to write that down, and then I'm going to insert a picture for it as well. So for instance,
So that would be that everything across that forward reverse would all be 0.
yes. So if they're all negative results, so it's with, so I won't write in the anti-A, anti-B because I'm going to show you all in the picture here in a minute. So all negative results with a negative autocontrol. Could be a group O, newborn or elderly patient. Let's see if it's this one. Yeah, here we go. Oops. Are y'all still able to read this? So your forward grouping would be anti-A negative, anti-B negative, which means they don't have any of the antigens, so they type as O. But then the reverse grouping should be positive and positive, but their reactions with the A1 cells and the B cells are both negative. And I suppose that could be due to their decreased immune system not having the strength, but so note the absence of agglutination with reagent cells in the reverse typing is because the production of ABO antibodies can be weak or absent in the elderly. So you can check the age of the patient and then increase incubation time to 30 minutes. But note that that's not appropriate if it's a newborn. And then lower the temperature to 4 degrees Celsius for 15 minutes. Include O cells and the autocontrol.
All right, so maybe I'm going to ask a crazy question, but incubation,
That's putting it in a heat block, and it increases the reaction capabilities.
Okay. I guess because we hadn't done it in lab yet.
Yes, no, the incubation enhances the reactivity.
Okay, so
in most cases.
you just increase it a little bit with the elderly, and then you lower, but not with the newborns.
Yes, so you could incubate it for 30 minutes for the elderly, but don't do that for a newborn sample. So you would have to check the age of the patient first.
Okay.
If you had results like this, you could see, okay, that's a discrepancy. I need to see what's going on. So then what you would do next could be check the patient's age and see if they're old or young first, because you're going to get a lot more elderly or newborn patients than generally having these weird, either transfusion reactions or auto antibodies or, some of the reasons for your other discrepancies. So I think that's definitely a good thing to check first, given an example like this. And then just a little overview of inheritance patterns. So, for example, you'll have ABO, and then H, and then your SE. So your SE genes are located at 3 separate loci. So now your ABO, we know those are just for your ABO antigens. And then you've got H. And what is H code for? What does that influence?
Your precursor.
Uh-huh. What does it allow those sugars to stick to create? Is it the red blood cell membrane or is it your fluids?
Wouldn't it be the red blood?
Exactly. Yep. So your red blood cell membrane. And then you've got SE. And what does that have to do with?
Ah. The secretor. I should know that.
Yep.
FE
So
secretor.
yes. So secretor or non-secretor. And where is that going to be located?
In the fluids.
Exactly. Now let me see if I can put this nice little picture here again. Oh, and then your H is going to be, you'll get either big H or little h. And it's an amorph, and so are your ABO. Amorph.
So your little, how is it amphoric?
Let me find it for you. Let me see if I can share this. Give me just a moment.
Okay, you're right.
So your amorph is a mutated allele that has lost the ability of the parent allele to encode any functional protein. So an amorph mutation is a loss of genetic information for the synthesis of appropriate mRNA. That gets a little too in-depth.
So it's basically saying it doesn't have the ABO antigen on the secretor.
You have to have your you have to have the SE gene to be a secretor.
Okay.
And so these are you've got either big H or little h, and these are alleles.
Yeah.
And then your SE is going to be your alleles. And then your AB or O What are those also alleles?
Yeah.
Correct. And I'm going to put A and H, and then the next one will be B and H, or H by itself will be group O. And they're all alleles. And so I want to add that little picture again. Let me see if I can make it. So your ABO, your H, and your SE, are those overall, are those going to be your alleles or on a loci?
They're going to be your alleles.
Your individual ones will be alleles, but your loci is going to be your ABO, H, SE location. So like I said before, that's kind of like your, that's your loci is going to be your coffee shop, or your locus is going to be your coffee shop that you go to every week. And then your alleles are going to be what drink you get when you go there. So for ABO, you're either going to be group A, B, O, or AB. So whatever blood group antigen you get, and then it'll be either, you know, your big H, little h, or big S, little e, or little s, little e, are all going to be your alleles.
So that little line there would be your alley.
Nope, that would be your like loci. So where you find it, and then I'm going to put in the alleles right now. that make sense?
Yeah. So wherever your location is, that's where your alleys are going to be attached to.
Yes. So the location's going to stay the same for you, me, Lily, whoever, and but our alleles will be what's different. So they have more variance. But like, you know how when we talk about the Philadelphia chromosome in hematology?
A little, yeah.
So that the loci for that chromosome is going to be the same for anybody who has it. But then the alleles vary.
So the alleles vary due to what blood type you are then?
Uh-huh. So your ABO loci will be right there every single time. But, you know, from you to me to someone else, the alleles will change because someone's going to be group A and someone's going to be group B and someone's going to type as group O and someone will be AB. But those antigens are all at the same loci, but they will be different alleles. So your alleles would be whether you have A, B, or O, or I guess in that case H, or AB, but they're all going to be at the same location, no matter which ones you get. And then I didn't talk about subgroups on the first video. So subgroups, if a weak subgroup is missed, oh, in the donor too, I need to include that. So if a weak subgroup is missed in the donor, what happens? Do y'all know? For your ABO subgroups, if a weak subgroup is missed in the donor, what could happen?
Couldn't it cause some type of reaction?
It can. So what could happen is the wrong blood type could be given to the recipient, which would in turn cause some kind of transfusion reaction. So I'm going to start off with subgroups of A. So I want to make the note that A1 and A2 both react positively with anti-A. So what you're going to do is use that lectin dolichos biflorus. Use lectin dolichos biflorus. And that is also called anti-A1 or anti-A1 lectin.
I'll be right back. I'm just going to run her outside before she pees on my bed.
Okay. So you use the lectin dolichos biflorus, which is anti-A1 or anti-A1 lectin, to, what does it help? Or what do you tell the difference in? So that's how you tell the difference between A1 and A2.
So you have to use that lectin in order to tell whether it's anti-A1 or the anti-A2.
Correct. Now, positive or agglutination. Oops. So positive or agglutination what do you think that result is going to be? So if it's an anti-A1, is it going to be, and it's positive, is that going to be A1 or A2?
Wouldn't it be both? Because it says both react to a positive anti-A.
That's anti-A. So this is specifically anti-A1 or anti-A1 lectin. So anti-A is going to be different than your anti-A1. What is agglutination?
It's where it actually breaks up. When it's one whole cell, there's no agglutration. But in the tube, when it breaks up, that means it's
Well, your agglutination is going to be a positive antibody antigen reaction. So you have the antibody you need and the antigen you need, and they're reacting. So if you have an antibody to A1 or like your anti-sera, which is your antibody for A1, and you have a positive agglutination, that means you have an antigen to what?
A1?
A1. Yep. It's just like forward typing.
Wow.
So your positive is, oh, sorry, Michelle, what?
I don't understand that.
Okay, let me write out this one first. So negative or no agglutination is going to be A2. And I will explain. So we talked about your anti-seras. So agglutination agglutination equals antibody plus antigen reaction. Well, that's not what I wanted. Just a minute. Okay, so your agglutination is antibody and antigen reaction. So if you have your anti-A1, so if you have an antibody to A1, to get a positive reaction, you have to express the antigen for A1 also. So if it's talking about A1 and you're getting agglutination, which means you have the antigen too, that means that it's going to be positive for A1.
Okay.
Does that make sense, why?
A little, yeah.
Okay, so like for your forward typing, you're using anti-A and anti-B. So if you have antigens with anti-A, if you have the antigen on your red cells, anti-A will react with the antigens on your red cells. So whichever one you react to is what you group as. And it works the same with the anti-A1.
Oh, so on the chart here where it says anti-A and A and B cell, is that what you're talking about?
No, this is the subgroup. So
Oh.
the forward typing is anti-A and anti-B. And then
Right.
this is a subgroup of A. So that's why I'm saying A1 and A2, both subgroups, react positive with anti-A. So the anti-A you use in forward typing, both of them react positive with it. But
Okay.
to tell the difference in them, you use this anti-A1. And if it's positive, that means it's anti-A1. And if you get a negative reaction, that means it's A2 because
Okay.
it's not reacting with the anti-A1.
Okay.
Now, 80% of individuals are A1. Out of your A and AB groups, and then a rare subgroup. If you have a rare subgroup present, your anti-H will be a strong reaction, and then your anti-A1 will be present, and you'll have weak or no agglutination with anti-A or anti-AB. And then I will put a picture up here in just a second. And let me put one more comment too. I'll put the picture in, but I'll put the comment up here because that way it's not below the picture and it doesn't get confused. So anti-A1 is not clinically significant other than incompatible cross-match. Let's see which one. I guess I didn't put down that one. Let me see if I can find a better example of just other than just saying. Just a second, I'm still looking for a picture for those results. Here we go. Let me take a picture of this and email it real quick. Okay, give me just a moment. I sent that. Okay, I'm going to give that a minute to go through, and I will come back to the rare subgroup present, what that would look like lab test wise. So I will come back to that in just a moment, but for now, I'll go on to subgroups of B. Now, these subgroups of B are rarer than A subgroups. They typically don't react strongly with anti-B reagents. And again, you can look in your book and find the results for like your subgroups of B, but you're just not gonna see it that often. Okay, I'll give that another minute or two to send. The next part is going to be our transfusion regarding ABO. So ABO and transfusions. So your red blood cell transfusion. So your ABO identical is preferred. And what does that mean? ABO identical.
Meaning they're compatible with all three.
Well, this one's actually, so ABO identical is preferred. So if someone has type B positive, they would rather them have B positive. But if B positive is not available, then next you can do ABO compatible. So an ABO compatible for B positive would be O negative or O positive. That's not identical, but it's compatible. Oh, but if you are transfusing whole blood unit, it has to be identical. Incompatible, what does that lead to? If you have incompatible blood that you are transfusing into someone else.
It means it's not going to take because they're different. It's going to react.
Yes, so it causes transfusion reactions. And note that acute reactions are often deadly. So who's your universal donor?
Oh,
Yep, so group O is your universal donor. And so who's your universal recipient?
AB.
Yep, so group AB is your universal recipient. Yeah.
what was AB again?
Your universal recipient. So here's your compatibility chart. And note that a lot more goes into it than just this. So there's storage, there's QC, expiration, units, what kind of units they are, how they're being kept, what temperatures they're being stored at, all kinds of different things that get included in any kind of transfusion practice.
And
But
this is in our book, right?
yes,
Okay.
and so if you have group A, for whole blood, what's special about whole blood transfusions?
They require identical ABO.
Yes, they are required when you're transfusing whole blood. So identical matches are required. So group A can only have group A whole blood, group B can only have group B whole blood, group AB can only have group AB whole blood, and group O can only have group O whole blood. So that has to be an identical match right there. But your donor cells, group A can have group A or group O for red blood cells. For group B, they can have group B or group O. And then your group AB can have AB, AB, or O, so any kind. And then your group O can only have group O cells. And now your plasma is opposite of that. So group A can have group A or AB plasma. Group B can have B or AB plasma. Group AB can have only group AB plasma. And then your group O can have O, A, B, or AB. So it's the opposite of.
So on the group AB, they have to have the exact same, they can't have just A or B. The plasma part, it has to be the same as theirs, which is AB.
Correct. So in the case of plasma, these get switched. So group O is a universal recipient and group AB is a universal donor. So for plasma transfusions, again, ABO identical is preferred, and ABO compatible is next. But that's when you keep in mind that it is reverse of the ABO compatibility. or the red blood cell compatibility. And I can't type today. So what that means is the donor unit antibodies must be compatible with the recipient red blood cell. So donor unit antibodies must be compatible with the recipient red blood cell. So in this case, your group AB, are they going to be the universal donor or universal recipient?
Universal recipient? Donor.
Donor for plasma, because these are talking about plasma.
Donor.
Yep, and so then group O are going to be your what?
Donor.
Gonna be what?
Recipient. Recipient.
Yep. These your plasma transfusion can be used for coag deficiencies. Is one use for them. And then again, make sure you know the difference between compatible matches for donor red blood cells and donor plasma. And then know that donor whole blood has to be what?
Identical.
Identical. And now let me come back and add in my A subgroup picture. Okay, so here would be your example of your A subgroup reactions. So if you have A1 versus A2, if you have A1 it'll be, they'll both be, A1 and A2 will both be positive for anti-A, negative for anti-B. And then for anti-AB, they'll both show up positive, except for with anti-A1, only your A1 will be positive.